2.1 Sensation vs. Perception. Sensation aligns with transduction. Transduction is the conversion of physical, electromagnetic, auditory, and other information from our internal and external environment to electrical signals in the nervous system. Sensation is performed when receptors in the peripheral nervous system forward the stimuli to the central nervous system in the form of action potentials and neurotransmitters. Sensation is the raw signal that is unprocessed and unfiltered until it reaches the central nervous system. Perception is the process of information to make sense of its significance. Perception occurs in the external sensory experience and the internal activities of the brain and spinal cord. Sensory receptors. Sensory receptors are neurons that respond to stimuli and trigger electrical signals. Distal stimuli are stimuli that originate outside of the body. Proximal stimuli are stimuli that directly interact with and affect the sensory receptors, and inform the observer about the presence of distal stimuli. Sensory receptors can encode many aspects of a stimulus. Psychophysics is the study of the relationship between the physical nature of the stimuli and the sensations and perception they invoke. Ganglia are collections of neuron cell bodies found outside of the central nervous system. After transduction occurs, the electrochemical energy is sent along neural pathways to various projection areas in the brain. Some types of receptors include the photoreceptors, hair cells, nociceptors, thermoreceptors, osmoreceptors, olfactory receptors, and taste receptors. Photoreceptors respond to electromagnetic waves in the visible spectrum to help with sight. Hair cells respond to movement of fluid in the inner ear structures to help with hearing, rotational and linear acceleration. Nociceptors respond to painful or noxious stimuli to allow somatosensation. Thermoreceptors respond to changes in temperature to allow thermosensation. Osmoreceptors respond to the osmolarity of the blood to allow water homeostasis. Olfactory receptors respond to volatile compounds to allow smell. Taste receptors respond to dissolved compounds to allow taste. Thresholds. Threshold is the minimum amount of stimulus that renders a difference in threshold. Some types of thresholds include absolute threshold, threshold of conscious perception, and difference threshold. Absolute threshold. Absolute threshold is the minimum of stimulus energy needed to activate the sensory system. This is a sensation threshold, not a perception threshold. When talking about an absolute threshold, we are talking about how bright, loud, or intense a stimulus must be before it is sensed. Threshold of conscious perception. It is possible for sensory systems to send signals to the central nervous system without a person perceiving them because the stimulus may be too subtle to demand attention, and may last too briefly in duration for the brain to fully process the information. Another word for threshold is limina. Subliminal perception or threshold of conscious perception refers to the perception of stimulus below a given threshold. Discrimination testing or psychophysical discrimination testing is a method to analyze the limit in human perceptive ability. During this test, participants are presented with a stimulus that is slightly varied and asked to identify if there is a difference in the second stimulus. The difference between the current stimulus and the original is increased until the participants report noticing a change. Note that a stimulus below the absolute threshold will not be transduced because it never reaches the central nervous system, while a difference below the threshold of conscious perception does arrive at the central nervous system, but does not reach higher-order processing regions that control attention and consciousness. Difference threshold. Difference threshold or just noticeable difference refers to the minimum difference in magnitude between two stimuli before one can perceive this difference. Weber's law states that there is a constant ratio between the change in stimulus magnitude needed to produce a just noticeable difference and the magnitude of the original stimulus. For high-magnitude stimuli, the actual difference must be larger to produce a just noticeable difference. Weber's law appears to be accurate for all sensory modalities, except at extremely high and low ends of each range. Signal detection theory. Signal detection theory is the concept of the perception of stimuli that can be affected by non-sensory factors such as memory, motives, expectations, and experience. Signal detection theory focuses on changes in our perception of the same stimuli depending on psychological and environmental concepts. Signal detection theory helps to describe response bias. Response bias is the tendency of subjects to systematically respond to a stimulus in a particular way due to non-sensory factors. Catch trials are trials in which the signal is presented. Noise trials are trials in which the signals are not presented. In these trials there are 4 possible outcomes of hit, miss, false alarms, and correct negatives. Hits refers to the subject correctly perceiving the signal. Misses refer to the subject failing to perceive a given signal. False alarms refers to the subject seemingly able to perceive a signal when none was given. Correct negative refers to the subject correctly identifying that no signal was given. Significant proportions of misses or false alarms indicate response bias in the subject. Adaptation. Adaptation changes our detection of a stimulus over time. Adaptation can have physiological or sensory components and a psychological or perceptual component. Adaptation is one way the mind and body tries to focus attention on the only most relevant stimuli. Adaptation usually picks up changes in the environment around us. 2.2 Vision. Vision is a highly adapted sense that allows us to sense brightness, color, shape, and movement, and then to integrate these information pieces to create a cohesive 3D model of the world. Structures and function of the eye. The eye is used to detect photons of light. Sclera is the exposed portion of the eye, that is the white of the eye. The sclera does not cover the cornea. The eye is supplied with nutrients by the choroidal vessels and the retinal vessels. The choroidal vessel is a complex blood vessel between the sclera and the retina. Retina contains photoreceptors that transduce light into electrical information that the brain can process. Cornea is a clear dome-like window in the front of the eye that gathers and focuses the incoming light. Anterior chamber lies in front of the iris. The posterior chamber lies between the iris and the lens. The iris is the colored part of the eye composed of the dilator pupillae muscle and the constrictor pupillae muscle. The dilator pupillae opens the pupil under sympathetic stimulation. The constrictor pupillae constricts the pupil under parasympathetic stimulation. The choroid is continuous with the iris and the ciliary body. The ciliary body produces aqueous humor. Aqueous humor exists in the front part of the eye and drains into the canal of Schlemm. The lens is located behind the iris and helps to control the refraction of incoming light. Contraction of the ciliary muscle occurs under parasympathetic control. When the ciliary muscle contracts, it pulls on the suspensory ligaments to change the shape of the lens. Accommodation is the changing of lens shape. The vitreous humor is transparent and supports retina shape. Retina is located at the back of the eye, and acts like a screen consisting of neural elements and blood vessels. The retina converts incoming photons of light to electrical signals. The retina is part of the central nervous system and is developed as an outgrowth of brain tissue. The duplicity or duplicity theory of vision states that the retina has 2 kinds of photoreceptors, those that are specialized for light and dark detection or rods and those specialized for color detection or cones. Cones are most effective in bright light, and are used for color vision and fine sense details. There are 3 types of cones, each of them are named for the wavelength of light they best absorb. Short or S cones range from 400 to 550 nanometers. Medium or M cones range from 450 to 650 nanometers. Long or L cones range from 500 nanometers to 700 nanometers. Rods make up most of the retina and are more functional as they only allow sensation of light and dark due to a pigment called rhodopsin. Rods have low sensitivity to details and are not involved in color vision, but night vision. The macula is the central section of the retina that has a higher concentration of cones, and at the centermost of the macula is the fovea which only contains cones. As you move away from the fovea, the concentration of rods increases while the concentration of the cones decreases. Visual acuity is best at the fovea. The blind spot is where the optic nerve leaves the eye because there are no photoreceptors here. Bipolar cells, ganglion cells, horizontal cells, and amacrine cells are located between the rods and cones to the optic nerve. Rods and cones connect with bipolar cells which synapse to ganglion cells which connect to the optic nerve. Information is transmitted forward towards the lens from the rods and cones during the integration steps until the signal reaches the ganglion cells. Since there are many receptor cells in comparison to the number of ganglion cells, there is a loss of details from the receptors. As the number of receptors that converge through bipolar neurons onto one ganglion cell increases, resolution decreases. Therefore, color vision has greater sensitivity to fine detail than black and white vision does. Amacrine and horizontal cells receive input from multiple retinal cells before information is passed to ganglion cells. The amacrine cells accentuate slight differences between the visual information in each bipolar cell to help with edge detection and increase our perception of contrast. Visual pathway. Visual pathway is the physical anatomical connections between the eyes and the brain and the flow of visual information along these connections. Basic visual pathway begins in eyeball and optic chiasm, then lateral geniculate body, then the optic tract. Half of the visual field are contralateral. Left visual field is processed by the right hemisphere and right visual field is processed by the left hemisphere. There are 4 visual pathways: the left visual field, the left temporal field, the right visual field, and the right temporal field. Left visual field or left nasal field starts with the left eye, then lateral geniculate body that synapses to another nerve in optic tract leading to occipital lobe. Left temporal field or the most left of left begins in the left eye and cross over the optic chiasm to synapse at the right lateral geniculate body, then the optic tract, then the occipital lobe. Right visual field or right nasal field starts in the right eye, then lateral geniculate body, then optic tract, then occipital lobe. Right temporal field or most right of right starts in the right eye and cross over the optic chiasm to synapse with neurons in left lateral geniculate body, then the optic tract and the occipital lobe. The lateral geniculate is located in the thalamus. Some visual pathways inputs into the superior colliculus, of which controls some response to visual stimuli and reflexive eye movements. Processing. Processing refers to our ability to make sense of visual stimuli that enables us to interact with our environment. Parallel processing is the ability to simultaneously analyze and combine features regarding color, shape, and motion. During parallel processing, picking out the information regarding color, shape, and motion is feature detection. Cones are responsible for color vision. Parvocellular cells detect shape and have very high color spatial resolution. This spatial resolution allows us to see fine detail. Parvocellular cells have low temporal resolution, so they only work with stationary or slow-moving objects. Magnocellular cells have high temporal resolution but low spatial resolution, so magnocellular cells detect motion. Hearing and vestibular sense. The ear is an organ responsible for our sense of hearing and vestibular sense or rotational and linear acceleration detection. Structure and function of the ear. The ear is divided into the outer, middle, and inner ear. The outer ear includes the pinna or auricle that channels sound waves into the external auditory canal. The middle ear begins with the tympanic membrane of the eardrum. The eardrum vibrates at the rate of frequency of the sound wave. Louder sounds have greater intensity, which corresponds to increased amplitude of eardrum vibration. From the tympanic membrane, ossicles help to transmit and amplify the vibrations to the inner ear. The ossicles are made of the malleus or hammer, incus or anvil, and staples, or stirrup. The malleus is connected to the tympanic membrane. The staples rest on the oval window of the cochlea. The middle ear is connected to the nasal cavity by the eustachian tube. The eustachian tube helps equalize pressure between the middle ear and environment. The inner ear sits in the bony labyrinth. The bony labyrinth is made up of the cochlea, vestibule, and semicircular canals. These structures are continuous and filled by a perilymph suspended membranous labyrinth. The membranous labyrinth is filled with endolymph, a potassium-rich fluid. Perilymph can transmit vibrations from the outside world and cushions the inner ear structures. The cochlea is a spiral-shaped organ divided into 3 scalae. In the middle scale, there is the organ of Corti. The organ of Corti rests on the basilar membrane, a thin but flexible membrane. The organ of Corti is made of tiny hair cells bathed in endolymph. The tectorial membrane is a relatively immobile membrane that sits on top of the organ of Corti. The other 2 scalae are filled with perilymph and are continuous with the oval and round window of the cochlea. These 2 scalae are filled with perilymph. When sound enters the cochlea by the oval window, vibrations in the perilymph are transmitted to the basilar membrane, then to the round window. Note the hair cells in the organ of Corti are like the rods and cones of the eye because both convert the physical stimulus into an electrical signal that is then carried to the nervous system. In hearing, the auditory or vestibulocochlear nerve carries electrical signals to the auditory cortex. Vestibule. The vestibule is the portion of the bony labyrinth that contains the utricle and saccule. These structures are sensitive to linear acceleration, so they help with balancing and determining one's orientation in 3D space. The utricle and saccule are covered in otoliths. Otoliths are modified hair cells that resist motion when the body accelerates. Semicircular canals. Semicircular canals are sensitive to rotational acceleration. The canals are arranged perpendicular to each other and each ends in a swelling called ampulla. Hair cells are located in the ampulla. These hair cells bend when the endolymph in the semicircular canal resists motion due to head rotation. Auditory pathways. Auditory pathways in the brain are more complex than the visual pathway because most sound information passes through the vestibulocochlear nerve to the brainstem, where it ascends to the medial geniculate nucleus or MGN of the thalamus. From the MGN, neurons project to the auditory cortex in the temporal lobe for sound processing. Some information is sent to the superior olive for sound localization and also to the inferior colliculus for the startle reflex. Hair cells. Stereocilia are hair cells that are located in the basilar membrane of the organ of Corti. When the hair cells sway due to vibrations, ion channels open to cause a reception potential. Hair cells that are directly connected to the tectorial membrane help to amplify incoming sound. The basilar membrane changes thickness depending on location in the cochlea. Place theory states that the location of the hair cell on the basilar membrane determines the perception of pitch when the hair cell is vibrated. The cochlea is tonotopically organized. Other senses. Smell. Smell is a chemical sense and responds to volatile or aerosolized compounds. Olfactory chemoreceptors or olfactory nerves are located in the olfactory epithelium of the upper part of the nasal cavity. Chemical stimuli bind to their receptive chemoreceptors to cause a signal. Pheromones are smells that carry interpersonal information. Pheromones have debatable effects on humans but play a role in animal social, foraging, and sexual behavior. Olfactory pathway to the brain starts with an odor molecule is inhaled into the nasal passage and then to the olfactory nerves in the olfactory epithelium, then the receptor cells send signals to the olfactory bulb. From the olfactory bulb, the olfactory tract sends the signals to the brain and limbic system. Taste. There are 5 basic tastes of sweet, sour, salty, bitter, and umami or savory. Flavor refers to the complex interplay between smell and taste, which is affected by non-chemical stimuli like texture and the individual's mood. Chemical receptors for taste are different from olfactory chemoreceptors because taste chemoreceptors are sensitive to dissolved compounds. Taste buds are found in little bumps on the tongue called papillae. Taste information travels from taste bud to brainstem, and then ascends to the taste center in the thalamus before going through higher order processes. Somatosensation. Somatosensation is described in 4 modalities of pressure, vibration, pain, and temperature. Somatosensation has 5 types of receptors that are Pacinian corpuscles, Meissner corpuscles, Merkel cells, or discs, Ruffini endings, and free nerve endings. Pacinian corpuscles respond to deep pressure and vibration. Meissner corpuscles respond to light touch. Merkel cells respond to deep pressure and texture. Ruffini endings respond to stretch. Free nerve endings respond to pain and temperature. Transduction occurs in receptors and then travels to the somatosensory cortex in the parietal lobe. 2-point threshold is the minimum distance necessary between 2 points of stimulation on the skin, such that the points will be felt as 2 distinct stimuli. The size of the 2-point threshold depends on the density of nerves in skin area. Physiological zero is the normal temperature of the skin between 86 to 97 degrees Celsius. Temperature is a relative judgment. Objects under physiological zero are cold and objects above the physiological zero are warm. Pain perception results from signals sent from sensory neurons such as nociceptors. Pain relies on thresholds, so it varies from person to person. The gate theory of pain proposes that there is a special gating mechanism that turns pain signals on or off that affect whether we perceive pain. The spinal cord is able to preferentially forward signals from other touch modalities to the brain to reduce sensation of pain. Kinesthetic sense. Kinesthetic sense or proprioception is the ability to tell where one's body is located in space. The receptors for proprioception are found in muscle and joints and play roles in hand-eye coordination, balance, and mobility. 2.5 Object recognition. 2 types of psychological processes include bottom-up processing and top-down processing. Bottom-up processing or data-driven processing refers to object recognition by parallel process and feature detection. Top-down processing or conceptually driven processing is processing by memories and expectations that allow the brain to recognize the whole object and then recognize the components based on these expectations. Both types of processing is sufficient by itself. Perceptual organization refers to the ability of these 2 processes to be used in tandem with all of the other sensory cues on an object to create a complete picture or idea. Based on the information available in terms of depth, form, motion, constancy, and other cues, we normally fill in the gaps using Gestalt principles. Depth perception uses both monocular and binocular cues that involve processing from either one or both eyes respectively. Monocular cues include relative size of objects, partial obscuring of one object by another, convergence of parallel lines at a distance, position of an object in a visual field, lighting, and shadowing. Primary binocular cues help with distinguishing slight differences in images projected on 2 retinas and the angle required between the eyes to bring them to focus. Form of object is determined through parallel processing and feature detection. Motion of object is perceived through magnocellular cells. Constancy is the idea that we perceive certain characteristics of objects to remain the same despite differences in the environment. Gestalt principles. Gestalt principles follow the idea that there are ways for the brain to infer missing parts of a picture when a picture is incomplete. Gestalt principles include law of proximity, law of similarity, law of good continuation, subjective contours, and law of closure. Gestalt principles are governed by the law of pragnance, which states that perceptual organization will always be as regular, simple, and symmetric as possible. Gestalt principles. Pragnance or good figure and simplicity. Such that every stimulus is seen as simply as possible and the easiest interpretation takes fewer cognitive resources. Similarity. Similar things are grouped together. Color is one measure of similarity, grouped into columns, but it could be shape, orientation. Good continuation. Connected points resulting in straight or smooth curves belong together. Lines are seen as following the smoothest path. This holds true even complex images. Common region. Elements in the same defined region tend to be grouped together. Proximity. Things that are near to each other are grouped together. Uniform connectedness. Connected regions of visual stimuli are perceived as a single unit. Meaningfulness or familiarity. Stimuli form groups if they appear familiar or meaningful even though they don't actually exist in the distal stimuli, top-down basis. Common fate. Things moving in same direction are grouped together.